There you go. Cool. Alright, so we're gonna get started really quick. Alright, so this time, so last time we talked about the more of like the molecular mechanisms of contraction and how these things and how like the functional subunits give rise to forces in the muscle and how those things are regulated, how the ATP cycling works, and pretty importantly how the sarcomere, as its length changes, how the tension develops. So in just like a kind of a review for this time, we're going to be talking more about the gross structural relationships, so how pennation angle and cross-sectional area work. But this stuff is still really important. So can anyone tell me what is, within each sarcomere, what is the thing that generates the force? So there's a certain structural relationship that occurs, and that's what generates more or less force. Basically, can someone explain this to me really quickly? Like the most basic way you can think about it.
The cross bridge, the power stroke. So as the actin myosin groups, well, the myosin binds when it's phosphorylated, that causes the power stroke to occur, and the actin filaments decrease in size or move.
Close. The phosphorylation is, so there's always ATP, and so what is actually causing the power stroke? It's sort of reverse to that.
Yeah.
What's the relationship between the ATP being there and the power stroke? Does it happen before or after?
ATP's already there.
Mm-hmm.
ATP.
Exactly. And then, right, so that causes the conformational change. So, what's the difference between, oh, I don't have a pointer. Okay, so I'm going to be on YouTube. So, alright, so what's the difference between this and this structurally in terms of the force generation? So there's like a certain amount of something that happens, and that tells you how much force is developed.
Overlap?
Right, exactly. So keep remembering this idea. Overlap is really important. Keep thinking about the sarcomeres. So we're going to move a little bit away from that in this lecture. Not going to see them as much, but they're always there, and that is the reason for the force development. But what actually happens in real life is not that you hold a muscle at a isometric position, so it's not always at constant length, but when you pick something up, it's at a it's like in an isotonic way. So roughly speaking, you lift a weight or something, your muscle is going to change length, but the force is the same. So throughout the movement. So how a lot of these studies have been done is, well, the characterization of muscle mechanics is actually more accurately represented by force versus contraction velocity. So at very high, what this is saying is that at very high forces, at maximal isometric force, you're actually not contracting quickly at all. So so that sort of makes sense. I'm pushing on this table, right? I'm pushing really hard, so there's a lot of force, but there's no velocity, there's no shortening velocity. Conversely, if you move really quickly or you have a lot of high velocity contraction, but you're not, the fastest velocity is going to occur when you have no force being applied to your muscles. So what this means is, later we'll look at power curves, so you'll actually see that even though you're exerting a lot of force, you're not actually moving anything, you're not doing any work, so you're not going to have any power at the highest force at the end of the curve. So in the middle of the curve is where you're going to see you're actually exerting your most power. So the most balance between velocity and force. Okay, so power is something we're going to talk about right now. So does that sort of make sense? The relationship between velocity, the actual shortening of your sarcomeres, and the force that you generate. We'll look at it more closely later in terms of the overlap. You know what, we can just talk about it now. So when you're contracting your muscles really quickly, you have a lot of basically the velocity between your actin and myosin, they don't really see each other as quickly. So what that means is you're not going to have time for them to bind because there's a certain amount of binding time required. So when you contract really quickly, you're actually going to have lower force because you're going to have lower opportunities for each binding per given unit of time. All right? So why does this happen? We just sort of talked about the actin-myosin cross-bridging. All right, so when you start moving really quickly, your sliding velocity goes up, the cross-bridging goes down, and as a result, the force generation goes down. Conversely, if you give your actin-myosin time to bind, you're going to increase the cross-bridging. The cycling is going to be more efficient in terms of the number of binding partners, and the force is going to go up. Does that make sense? Questions? Good. Okay, so power is force times velocity. So sort of like, so when I talked about before the relationship, the force velocity curve, this is where it starts to matter in, I guess, in real life. How much of that force can you exert in how much time? And it's sort of at the middle of the force velocity curve. It's just really, you just transform the other graph by multiplying their, basically you change the scales. The same scale, you transform basically. And then you start to see that there's an optimal length in the middle. So we'll see how this changes when you start changing different parameters of forces, weights, and cross-sectional area. So imagine you have two people, or you can think about one person changes their velocity and they're performing some action, in this case going downstairs. So you change their velocity, and how does that affect the power curve? Can someone take an educated guess on how they think that the power curve would look like if you decrease the velocity for man A versus man B? How do you think that the shape of that thing will change if you change one of the values of force versus velocity? Based on like the multiplication.
Since power is equal to, power equals to force times velocity, any of those decreases, power is going to decrease.
Right. So the
Curve
shape.
is gonna
Right. Okay.
lower.
Exactly. Okay. So that's what we see. If you decrease the velocity, you're going to have the lower power curve, but I think the shapes are the same. I mean, the shapes should be the same. The maximal power will decrease. And you can see that the where's my cursor? You can see that the velocity here is there's a large difference between the force velocity curves, and then that translates to the power curve is decreased for man B. All right, pretty self-explanatory. So now we change the amount of force involved, or in this case, we're going to change the mass, but we're going to keep the velocity the same. So again, we're going to change the amount of force you see. So does anyone know, just quick physics question, where does the, why is it 981 watts? Sorry?
Because of gravity.
Okay, great, good, fantastic. So, I'm just trying to get some interaction. Alright, so yeah. Again, if you decrease the, force in this case, you change the weights involved, you're going to decrease your power curves. Reflected this time in it decreased in the force, but the starting velocities, or I guess velocities, are the same. All right. Okay, so this is, I'm going to be referring to these little areas where you see parallel versus pinnate fibers a lot, a couple times in the lecture, so keep these in mind. So from last lecture, cross-sectional area is the amount of area over which the work is being performed, I suppose. As this area increases, the force production increases. And another important parameter is the fiber length. So the longer that each fiber is, the more sarcomeres in series. So could someone tell me what's the benefit of having, or the pros and cons of long fibers?
Greater velocity.
Okay. And why? How does that?
Because the fiber is longer, so there are more sarcomeres, and they're faster to I don't know how to say this, like to overlap.
Right. Okay. So they slide. That's right. So is there an actual difference between each sarcomere? Like if you have a longer muscle length? Okay. So there's no difference. Great. So, all right. So can someone tell me the benefit of having the box on the right, pinnate fibers? Read it up. It's not hard. Good job. Extra points.
There's more power in the muscle because the cross-sectional area is bigger than in a fusiform muscle.
Close. We're just going to talk about force for now. So there's more force. All right. So we don't know about the shape of the graph yet, but we'll talk about that. So yes, that's right. There are more sarcomeres that are contracting any given contraction in a cross section. So the cross sectional area is larger. Okay, so now we're going to play with the cross sectional area equation and see how these different when you start changing things like pennation angle, muscle density, fiber length, and muscle mass, how does that affect the behavior of the muscles? So right now we're going to take a look at changing the muscle mass, and when you keep everything else constant, doubling the muscle mass will result in a greater cross-sectional area. So that makes sense. So you have a larger muscle, you change your cross-sectional area, and then you change the resulting force. Okay? Any questions about how this works? the equation or anything like that. So it's not actually an area of like a cylinder or anything like that. It's there's more physiological parameters that are involved in that. Okay, so when you change mass, you change your cross-sectional area. So what happens to the length force curves or the power curves? So like we just talked about, when you change the cross-sectional area, you change the force. So at any given length, you're actually going to have an increased force for a higher cross-sectional area. So when you change the, when you look at the force-velocity curves, you actually have increased power because you're going to be increasing that force measurement in the multiplication that we talked about before. So at any given time, when you're doing contraction, you have a greater number of sarcomeres, greater force, more power. And how does that look like? Well, roughly speaking, you just take the in parallel, you would take more of those. You don't change the density, you don't change the angle, so all you'd have to do is increase, roughly speaking, a cross-sectional area, and then that is sufficient to give you a higher force. Pretty intuitive, I think. Okay? So now, what happens when you have different fiber lengths? You'd actually have to change the masses to keep these things constant, but when you have different fiber lengths, could someone take a guess on how the force, velocity, power curve would look like? All you're doing is changing the length of the fiber.
Gonna say either it's either gonna decrease or increase depending on longer or shorter.
Mm-hmm. So increasing the length of the fiber.
Yeah.
So
So increasing, you're probably gonna decrease. Oh well, if it's okay, if I'm looking at cross sectional area, that would decrease that. But I'm just looking at the equation.
Mm-hmm. So no, I'm asking. Yeah. So we keep the cross sectional area constant.
Okay.
Okay, so imagine we keep the cross-sectional area constant, and we're trying to look at an effect of fiber length. So on power curves. So what would happen if you lengthen, if you have a longer muscle to the power that you can output?
Increase.
Yeah. So okay. So you would increase your power because which parameter changes when you lengthen the muscle? Mm-hmm. Okay. So when you lengthen the muscle, you increase the velocity because you have more sarcomeres in series. Just at any given time, they all shorten the same. Every sarcomere shortens in the same rate. You have an increased distance covered in the same amount of time as you would shorter sarcomeres. Okay? So we see that here. So the velocity. For a longer fiber, you see that the velocity, starting velocity or the maximum velocity is different, and that is sufficient to have a higher power over the entire curve. Does that make sense? So when you're changing these parameters, just look at the axes, okay? So you see that the forces, so why are the forces the same based on what we just did to the when we looked at the equation? What is related to the force? Right. So we didn't do anything to the cross-sectional area. So we're not changing the the force output, but we did change the length. Okay, so we're going to change the power output along the length of the curve. So this makes sense as well, right? So why are these maximal? Why are the maxes of the curves the same?
Because their PCSA is the same.
Exactly. So we're just gonna keep going over this in like 60 different ways. Sorry. They have the same maximal force output. But this is done over. So they reach their maxes at shorter or longer lengths, and that makes sense because they have a longer operational. So longer fibers get to work over a longer distance. Okay? Here's my crappy slide. Okay, so this makes sense. Okay, so it's the same cross-sectional area, but now you just have longer fibers. Any questions? Great. Alright, so now we're going to change the pennation angle. Okay, so if you start to change the angle, what happens to the... Pretend you can't see the answer. What happens to the cross-sectional area when you start changing the angle, and tell me why. Tell me why after you give the answer. Yeah, so it's like a two-part kind of thing. What happens when you change pennation angle to your force output? You increase your force output. So you can stick more things within one of those boxes, and then eventually you have an increased cross-sectional area, and that's just based on the math of how this works, I suppose. And that makes sense because you have to change the force generation as a function of the degree. So how that changes your force velocity curves is that you have the same velocity, okay? So we're not affecting velocity here, but we're changing our final forces on our ending or starting forces, depending on which way you want to do the contraction. And then you have an increased power within certain parts of the curve. But because we're also changing, I think we changed the length. Yeah, we're also changing the length. So because this length is changing, you're having a different range of motion. So for these longer fibers, you actually have there's sort of like an inflection point at which the increased length is able to carry that force longer, because the other muscle in comparison will have already finished its range of motion is limited, right? So the total possible length over which something can work, it makes sense, is longer for a longer fiber, okay? And this is also reflected in the power curve, while a shorter muscle or pennate muscle on average is shorter, because within the box you only have two fibers which cover the length of the box, and then the rest of them are shorter, and then that averages out to a shorter less number of sarcomeres in series. You actually start to see that for something like a quadriceps, you have a very high force or a very high power generation in a shorter time window than something like your hamstrings, which act over have more power later on in the contraction cycle. Does that make sense? Any questions? Okay, so this is just a visual representation of how that works. As you change the angle, you necessarily change the cross-sectional area and at the same time another parameter, the average length. So not the length of these in total, but the population length. So you'd average these out and then you compare the length averages. Okay? So we're sort of changing two parameters at once here. Okay, so I sort of lied last time about how there are no differences between sarcomeres because there are some differences between fast contracting and slow contracting muscles. So so a lot of people have heard like sprinting athletes have fast twitch fibers and marathon runners have slow twitch fibers. I'm not exactly sure which one matters more, like the architectural things we talked about, or the fiber type. My sense is that architectural things matter more, but they all sort of work together to change your physiological properties. So if you just have someone who's like super type 1 or type 2 dominant, the effect is not going to be as large as someone who has different pennation angles in the muscle just because of the biophysics of it. But that is to say, there are definitely differences between the sarcomeres and the contraction times. And the sorry, I sort of went about that circularly or backwards, but what I'm trying to say is basically these are molecularly determined. And that is determined by the ATPase activity. So the actin-myosin interaction cycle occurs at a certain speed, and that changes whether or not you're in a slow muscle fiber, so type 1, or in a fast muscle fiber, type 2. And there are ways of detecting these things using histology or biochemically, and then imaging them to see whether or not you have fast or slow muscle fibers. So interestingly, in people, you can actually have multiple of these per muscle fiber. So you can have a mixed fiber, you can have type 1, type 2A, you can have type 2X, you can have all sorts of intermediates, and then these different isoforms or changes in the proteins can exist at the same time, and they're based on how closely they contract in terms of speed. So, and I switched that. So fast contracting fibers have a high ATPase, high turnover, and they generate a lot of velocity. So they're expecting very high speed differences between the actin and myosin, and then the ATPase has to be able to cycle through that or bind to the actin pockets to keep up with that speed of velocity, or the contraction velocity. So sprinters will have high ATPase activity. What's interesting is that sprinters actually have, based on our force velocity curves, do you expect a more quickly contracting muscle to be stronger or weaker? Someone said here? Fast contracting muscles should be weaker, right? So sprinters actually intrinsically sort of have weaker muscles, which is really weird. But so why do you think they're able to generate so much force? Even though their muscles are contracting really quickly. So what do they sort of look like? What do their muscles look like?
Maybe pinnate.
So we're assuming for the same muscle between like a marathon runner and like a sprinter. even though they're contracting really, they have really high ATPase activity, they're really quick muscles, they have maybe more type 2 fibers.
More muscle mass.
Exactly. So they're like huge damn legs. So they're actually kind of, which is weird, the actual muscle is a little bit weaker. So think about that. Think about the mechanics of how all this stuff works, but it's pretty interesting. They need more muscle to produce the same amount of, or to produce more force. So those little weaklings are actually pretty strong when you're looking at marathon runners. So how to find oxidative muscles, or things which are more the slower contracting muscles, as opposed to the ATP, the fast contracting muscles with high ATPase activity, or we can also look at oxidative potential. So oxidative potential is it's like the ability to produce ATP. Okay, so this is mitochondria produce your ATP, and so we're looking for something called succinate dehydrogenase, and this is in the inner membrane of the mitochondria, and it's intermediate in the Krebs cycle. And finding this, finding SDH, SDH activity is proportional to ATP production. And so, if you imagine a marathon runner, do you think they're going to have a lot of these, a lot of mitochondria or few mitochondria? And why?
A lot of mitochondria.
Okay.
Because they're oxidative fibers, so you're using
You need a lot of you need a lot of energy. So these fibers have high oxidative potential. So if you do if you look for these guys, they're going to have a lot of SDH activity. So you can distinguish these things together will help you make a profile, like in a cross section, which fibers are fast, which ones are slow. You can also look for glycolytic potential. So glycolysis, I'm going kind of fast. Is another way that cells can get energy, and this is predominantly done by the fibers that sort of don't have these ATP reserves, don't have as many mitochondria. So alpha GP, or alpha glycerol phosphate dehydrogenase, and PFK phosphofructokinase identify these high and low glycolytic potentials, and they're directly related to the rate of glycolysis. So muscles with high glycolytic potential have high alpha GP or phosphofructokinase activity.
So they found in sprinters, correct?
Yeah, so you'd more likely find these in the fast fibers. So based on what I said about mixed fiber typing, this stuff sort of gets a little bit hazy. So once you start, we should have added like a histology slide, but basically how this stuff looks like in cross section. Oh my god, I'm just such a good professor using the blackboard. All right. Okay, so that's your muscle. It's yours. Okay, and what happens if things were only type 1 or type 2, and we're looking for something like SDH activity, okay? So type 1, type 2. Okay, so type 1 has high SDH, and then we add like a dye, sort of, okay? And then this one has low SDH. It would be, this one would be So this one has a die, so let's say plus die. Guys sort of see what I'm getting at here? If you were to look for them, they'd be like this. Okay, let's pretend that it's like completely filled up. So these ones are your, which ones are these? Okay, right, so that's type 1. But in reality, what were we talking about fiber typing and what is actually called co-expression. So what actually happens? Is this like a realistic, do people only have this? No, right? So they have mixes. So what would you actually expect for this to look like with these mixed or co-expressions? So you actually have different isoforms in each fiber. So what I'm looking at is cross-section. Okay, so this cross-section. So this is actually okay, and we're just like looking at that. Okay? So what do you actually expect for this to look like? If you have intermediates, what changes to the shading? So what is the actual physiological, what would this look like? That's right. You have a you have actual shading. So these things exist. Oops. So there's a large spectrum of these, and they're all mixed. And so most muscles in humans are actually a mixture of these fiber types. So there are very few pure fiber type muscles. So if you have something like, but that's in humans. Humans we actually skew predominantly slowly, as opposed to something like rodents, which are they're way more fast twitch in total. And things like pigs actually have different, an extra fiber type total. So we have 1, 2A, 2X, pigs have 2B, rats have different fiber type compositions. So these things, the point I'm trying to make on all of this is that every muscle has a different fiber type composition, different percentage, which will affect its mechanics. All right, first point. Second point, this changes from animal to animal. This changes among people. Yes, go ahead.
But for our purposes, are we dividing these, or are we like, is it fair game to just for us to know about, like, will we know enough knowledge about when they're mixed together? Like,
No.
say for example, type 1 and then type 2, or type 1 and then type 2.
No. So I'm just trying to like, I'm just going. So what's not on the slides is not gonna. Okay. Don't quote me on this, but I'm 99% sure. Okay, that's a little bit. All right. If it's not here, you're not going to get tested on it. I'm just trying to explain how it actually looks like in reality, and I'm trying to tell you basically how these things work in real muscles. So we're going to just basically just go through the fiber types, and then I'm just going to tell you how they actually break down in real muscles, and then just think about it going forward. I don't know if it's interesting to me. It could be interesting to someone else, but in reality, these things exist on a spectrum, and it's pretty complicated how they look for these things, how they affect the biomechanics in real life. But this stuff, it's just to keep in your mind when you look at this in the future. It's so that they're just not like on your Excel sheet or something like that. Like there's a pretty wide spectrum, and it's not as clear cut as looking for these things as positive or negative.
But like, if we're continuing with that, is fast oxidative glycolytic, is that a mixture of the two, or is that just considered type 2?
So okay, so when you have the type 2X is the fastest one, and then the type 2A is intrinsically sort of in the middle.
Okay, that's fine.
And then you can have a type 2A, type 2X, which is in the middle of those two. So it's in the middle of fast and middle. So it's like middle plus, right? So you can also have type 1 mixtures with type 2A, etc. Okay, so based on what we just talked about, their oxidative and glycolytic potentials change between type 1, 2X, 2A. So it's low for the slow fibers. The ATPase is low. ATPase activity is low, and then it's higher for the type 2 fibers. SDH activity is going to be higher for the type 1 fibers, and lower for the relatively lower for type 2X, and I think it should be a little bit lower than the 2A, although here it says just high. And then you can go through it alpha, G, B, on your own. Okay, so the mechanical properties change between the type 1 and type 2 fibers. Like we talked about before, contraction velocity, or which is determined by the ATPase cycling, and also the T-tubule and sarcoplasmic reticulum system. So really large type 2 fibers are going to need a really quick way to transmit a signal. So they have what we talked about last time, the T-tubule system, which sort of wraps around, and it sends out, it spreads the action potential. So really large fibers need a quick way to do that. So the really large fibers are going to have a more reticulated TR system. So that allows for increased rate of muscle activation. Okay. And for the force power curves, again, what we talked about with the sprinters and the marathoners, the sprinters are going to have more of those. It's not that much more, by the way. I think it's only per muscle. It's only going to change by like 10 to 15% or something like that. So you can there's a big question whether you can actually train to have more type 1 or type 2, and how much of that is actually genetic heritability. You can change it in some ways, but yeah, I'm going off topic a little bit. But in any case, these guys have more power. Okay, the type 1s have lower power. And again, we're only changing, in this case, we're looking at velocity, so we're not going to change the force output right now. And again, talking about the complexity of how these things work. This is only when we're looking at the pure isoforms. So when each fiber only has one shade in it, as opposed to two mixtures of these things. But you can imagine that things with type 1, type 2 have a mixture of these properties. But when we start looking at things of like Vmax, force production, power output, you would expect all these things to be lower for type 1 fibers or slower fibers, and faster for type 2A and type 2X. The type 2As are going to be intermediate for most of these metrics in any case. So you can think about, when you're studying these things, you can think about clumping those together based on power and speed. So again, for type 1, you have higher oxidative capacity, lower glycolytic capacity. These things also have lipid droplets. So I guess, interesting fun fact, a lot of things that are, when you get older, the lipid droplets in your muscles actually start to get larger and larger. And if you really leave these muscles to just stay there and atrophy, you'll actually convert entire muscle cells into fat cells. So it's one of the rare cases where you actually have a change in your cell identity after development, which is actually kind of crappy because, yeah, you literally change muscle into fat. But that luckily doesn't happen so often, but often enough for it to look like, if you have ever seen like a scan of an older person's leg, you see that a lot of the inside of their muscle is marbled with fat, and that happens when you're completely inactive. So these lipid droplets are great for fuel storage, but over time, maybe not such a great thing. But for fast fibers, these lipid droplets don't exist, or there are very few. and in 2x there are almost none. But these fast fibrils need to have other forms of storage, so they have granules of glycogen, so they use that really quickly. So that results in a very short duration of use, whereas the lipid droplets can be used for a very, very long time. So marathon runners, of course, from marathons. And then you have someone like 400, 800 athletes, which are pretty quick, but sort of in the middle. Yeah, I think that's basically all I have to say about that. Are there any questions?
I have a question. With myosin phase activity, what is the benefit of that being low? Like, is that promoting efficiency in a slow pitch muscle fiber, or how is that helpful?
Okay, so the ATPase activity has to. Okay, so ATPase activity has to match the maximal ability of contraction. So if you. Okay, so what is a benefit of a slow contracting fiber in terms of force, velocity? Based on that, we can do this better. So remember the force velocity curve. What is the benefit of contracting slowly? You always make trades. So what's the trade for slowness?